All right, since it's past 12, let's get started. Let me know if you can't see what I'm writing or if something isn't isn't clear. Yeah, no worries. Let me send the link again. Also, maybe as new people are coming in, the link might not show for you. So what I'll do is at the end of the session, I'll put the link in the chat again. So people that come later can also fill out the link. So, all right. So today we're going to be doing a lot of conceptual and stuff from the textbook. So today, there's two worksheets. So if we have time, we're gonna finish diversification of eukaryotes and then get to prokaryotes versus eukaryotes. So in this activity, we're gonna see early evolution of eukaryotes, single cell organisms, and see what types of organism came first and how it evolved to the other. So a little bit of background. The first eukaryote appeared in fossil record around 2.1 billion years ago, and current evidence indicates that eukaryotic cells arouse as a result of endosymbiosis. And that word should sound familiar. It's where they have different relationship with other organisms, and sometimes it's even multiple endosymbiosis of prokaryotes. And the early eukaryotic cell that resulted underwent great diversification to become the 7 major groups of eukaryotes. And if you want picture reference, you can also look at the figure in the biological science textbook on page 561. So the first question we're going to look at is um, why prokaryotes are called catch-all groups, and what does that mean? So first we know that it's a paraphyletic group, and it includes um, all eukaryotes that aren't part of the fungi, animal, or plant kingdom. Also, I know that both the science professors said they'll put up the worksheet ahead of time for you guys, so if you want, you can get the worksheet and follow along with me as I'm writing these answers out. And another reason that why they're called the catch-all group is because they're just super diverse. So they vary greatly in size. They can go from macroscopic to microscopic. Some can be as long as 60 meters long, and they have characteristics like overall form and have distinct organelles, or sometimes even both. So they have range of size. And it's important to try to recall that all eukaryotes alive today, including these protists, have nucleus and endomembrane system, including the mitochondria and or genes that are normally present in the mitochondria and cytoskeleton. Okay, so for the second question, it says there are more than 100,000 recognized species of protists. Even though the organisms in this group are so diverse, we can recognize some common themes. Oh, looks like my pencil isn't working. Okay, so they have common themes in their evolution, including complex cell structures and novel genetic recombination strategies, and complex lifestyle. So in the previous question, we talked about how protists are very diverse and are called the catch-all group, but now we're going to be looking at their common themes and what stuff that they have in common, including their cell structure and how their genetic recombination strategies work, including their lifestyle. So for the next few parts, there can be multiple right answers. So if you look in chapter 27 in biological sciences, you can find a lot of different answers, but I'll just be going through a couple of the examples that I've pulled out. Okay, so the first thing we're going to be looking at is the theme of complex cell structure. So the first example we're going to look at is paramecium. So is anyone familiar with paramecium? Okay, so the cell structure of paramecium, it includes a contractile vacuole. It also has an oval groove and cell mouth. And it has micro and macronuclei and cilia. Another example of a protist. This protist has light detectors. It also has a contractile vacuole. Pyrenoids and chloroplast. So one of the comments says, are these types of protists found in the textbook? Yeah, they're found in the textbook, and there should be more examples of them too. And I'm just going over two examples for each of the section. But if you refer to the book, you can find more details about these examples and a couple of other examples as well. Okay, so the next theme that we're going to be looking at is novel genetic recombination strategies. So let's look at paramecium again. In the last section, I mentioned that they have a macro and micronuclei, so we'll be looking at what exactly those does. So the micronuclei is going to be helpful in sexual recombination. And the macronuclei is going to function in growth, maintenance, and asexual reproduction. So primarily involved with growth and maintenance. Another protist example is Laminaria. What this protist does is that it shows an alternating generation between the gametophyte and the sporophyte. Does anyone remember if the gametophyte is a haploid or a diploid stage? Okay, so the gametophyte is going to be the haploid stage, and the sporophyte is going to be the diploid multicellular form. So these are two examples of recombination strategies that different protists have. Yeah, haploid. See a couple comments saying haploid. Yeah, good job getting that right. Sorry, I think the chats are a little bit slower on my end, so it's coming back a lot later. But when I see it, I'll respond or comment. Are the alternating cycles for only multicellular protists? That's a great question. I'm not too sure about if it's only for multicellular protists. But the diploid form is a multicellular form. So I'm assuming that it might only occur in multicellular protists. But I would have to check on that. Great question, though. Okay, next, we're going to look at their life cycle. So first, we're going to look at Plasmodium. So these have a two-host life history and are also known to cause malaria. So it reproduces asexually in the human host, and it reproduces sexually in the mosquito host. So depending on the host, the protist can change what kind of life cycle that they're going to utilize. So that strategy is called a two-host life. And another example we can look at is the cellular slime mold. So what these do is that they have a life cycle that includes a single-celled feeding amoeba, and then in their sexual phase, which is the haploid, they can fuse to form a zygote, and then that immediately goes under meiosis, and that helps it like form into colonies. And once it forms into the colonies, then they can go through asexual fruiting, and that gives rise to the spores that you see when there's mold. And what they do is that the asexual reproduction, when they produce the spores, they usually utilize that when the environmental conditions aren't ideal. So um, so they want to utilize the asexual phase because it doesn't require as much energy as the sexual reproduction does. So these also have a sexual phase and an asexual phase. And asexual phase is favored in unfavorable conditions. Any questions so far? All right, let's look at some ecological importances. Can anyone just throw out ideas for why protists are important to the ecosystem and the environment? There's some great benefits of protists to marine and freshwater environments. Yeah, the comment says, I think I remember something about carbon. Yeah, that's kind of on the route that we're trying to go. So, absorbative and fungus-like protists aid in decomposition, and they recycle organic matter. Yeah, compounds. So that's one great ecological importance that they have. Another big importance that they have is certain fungus are photosynthetic. Um, when they have like cyanobacteria, and what that does is it provides food for the different ecosystems in freshwater and marine environments. So it's really important that we have those photosynthetic funguses, or protists, I should say. All right, let's move on to the next question. So we're going to be looking at endosymbiotic theory and the origins of eukaryotic cell. So can anyone tell me what the endosymbiotic theory is? Um, a hint could be that our mitochondria came to us through endosymbiotic theory. Comment says mitochondria originated when a bacteria cell took up resident inside another cell. Yes, perfect example. Um, and that is a great example of the endosymbiotic theory. And it says that eukaryotic cells originated when these ancestral prokaryotic cells um, engulfed other free-forming prokaryotic cells and just enclosed them into their own vacuoles. And this is how major eukaryotic organelles came about. And a perfect example is the mitochondria. So let me write that out. Yeah, so we already talked about one example, which is the mitochondria. So I'm going to write that down here. Does anyone know what the evidence that they use to support this theory for mitochondria? Why do scientists think that mitochondria was engulfed by a prokaryote? Can you guys think of any similarities between a prokaryote that the mitochondria has, even though it's inside a eukaryotic organism? Comment says can replicate itself through binary fission. Yes, that's a great example. As humans, we don't really do binary fission when we want to replicate. So that's a great example. And their DNA material is circular, whereas our genetic material is not circular. And that also presents another evidence for why it's a prokaryotic organism. So circular DNA. That replicates through binary fission. Yes, another comment says they have their own ribosome. Yes, that is a great answer. And another detail about the ribosome is that they have a 70S ribosomal subunit, which eukaryotes don't. We have a 80S subunit. So it's really interesting that they have a 70S. And just another observation that the scientists made is that the inner membrane protein of the mitochondria are actually more similar to the prokaryotes than eukaryotes. So that provided more evidence for why the endosymbiotic theory holds true for mitochondria. Can anyone give me another example of organelles that were produced through endosymbiotic theory? Yes, great job, chloroplast. So what are some evidence for why chloroplast came about? through this theory. All right, I'll just tell you guys because it's very similar to the last answers you gave me. So I'm just gonna do this, similar to above. So they also have their own circular DNA. They also have the 70S ribosome, and also in the chloroplast, they noticed that the inner membrane proteins are more similar to prokaryotes than eukaryotes. All right, any questions so far? Okay, let's move on to the next section. Okay, so the chloroplasts of some organisms are thought to have arisen as a result of secondary endosymbiosis. Does anyone know what secondary endosymbiosis is? Okay, so previously we talked about how a prokaryote engulfs another prokaryote and then forms a vacuole around it, and that became the endosymbiotic theory that we just looked at. But the secondary endosymbiosis is said to occur when heterotrophic eukaryotes engulf a photosynthetic eukaryote, and then those become the endosymbiont. So secondary endosymbiosis is heterotrophic. The big difference here is that they're both eukaryotes here. And then some of the evidence for the idea of secondary endosymbiosis is that the chloroplast is surrounded by four membranes and it carries out photosynthesis and has its own plasmids as well. So usually it's a double membrane, right? So because it has four membrane, you know that the engulfing happened twice. That's why it has four membranes now. Okay, structurally, Giardia lamblia lacks complete mitochondria and was once thought to be an example of what the earliest living eukaryotes may have looked like. If Giardia is similar in structure to the early living eukaryotes, what does this imply about the order of evolution of the various eukaryotic organelles? So what do you guys think? So based on this information, which one do you think evolved prior to the endosymbiosis? Do you think the Giardia structure came after endosymbiosis of mitochondria or chloroplast occurred or before? Okay, so the question tells us that they lack, um, comment says before. Yes, yes, that is exactly right. So the question says that, um, yeah, so the question here says that it completely lacks mitochondria. So that's evidence that this organism lived before endosymbiosis ever occurred. Okay. We have since discovered that diplomons and the parabacillids have modified mitochondria. What type of mitochondria do they have? So they say that they appear to be vestigial mitochondria because it appears unusual and contains genes, but still contains genes that are normally found in the mitochondria. So it is said to be vestigial mitochondria. And what this observation indicates is that it supports that ancestors had mitochondria, but then the organelle was lost or reduced over time. And that supports why vestigial mitochondria can also be seen. Okay, next we're gonna try to determine what type of organism came first. So one is going to be the earliest and five is gonna be the latest. So I'll give you guys a couple of minutes to look at the answers that are given and just throw in um, which one you think came first or which one you think came second. How I would go about answering this is first determine if you think eukaryotes came first or prokaryotes came first. So what do you guys think? Do you guys think eukaryotes evolved first or prokaryotes evolved first? Yes, that's exactly right. So prokaryotes came first, so I would momentarily ignore all the answers that says eukaryotes. And then it says, so the second half of the question is talking about what type of cellular respiration mechanisms they have. So it says can perform Krebs cycle, electron transport, photosynthesis, or glycolysis. So which one of these mechanisms can occur without oxygen? So Krebs cycle is indirectly going to require oxygen. Photosynthesis is going to require oxygen. Yes, glycolysis. Um, that is the one that doesn't require oxygen. So this combination, the prokaryotes and the glycolysis one. So that seems to be the most primitive one. So we can go ahead and say that's what evolved first. So we know glycolysis occurs in the cytoplasm and can occur in the absence of oxygen. And because we know that all eukaryotes came from endosymbiotes, it's a good guess that the oxidative respiration came next because you need another organism to be able to do electron transport chain. So using the similar logic, the next thing that evolved are prokaryotes that are capable of performing photosynthesis, and also prokaryotes that are capable of performing electron transport. So kind of around the same time. But it's important to know that this is what came first. And then next in the evolution, what happened was prokaryotes capable of performing Krebs cycle, and then it's eukaryotes capable of performing Krebs cycle, and simultaneously eukaryotes capable of performing electron transport chain. And then finally, um, we have eukaryotes performing photosynthesis. And this again goes back to like the double symbiotic thing that happened where there's cyanobacterium that has chloroplast and then that gets engulfed by the photosynthetic eukaryotic cell. Okay, so that takes us to the end of this worksheet. The next worksheet that we have are going to look at the difference between um, things from different kingdoms. So before we move on to that, any question regarding this worksheet? And these videos will also be posted on my Twitch channel and it'll also be on YouTube later this week. So you can always rewatch it if you need anything to be slowed down. Okay, let me get the other worksheet set up. Okay, so in this worksheet, for the remaining of the time, we're going to be looking at the main characteristics of bacteria and archaea and eukarya. So let's first look at bacteria. Can anyone tell me some key characteristics of bacteria that make them unique? I know we already mentioned their circular DNA before. Um, What about their cell wall? Can anyone tell me something about their cell wall? What it's made out of? Comment says, was there a form to fill out at the beginning of the session? Yes, there is, but I think when people come in after I put in the link, it disappears. So what I'll do is at the end of the stream in 20 minutes, I'll put up the link again for people that are here and you can fill it out to get the extra credit. Okay, so the cell wall of bacteria has peptidoglycan. And this is the feature that we use to determine whether something is gram positive or gram negative. So the presence of a large peptidoglycan cell wall is said to be gram positive, and the absence is gram negative. They have lateral gene transfer, no membrane-bound organelles. Yes, those are some great responses. And peptidoglycan, yes. And also, all bacteria use the same type of RNA polymerase. Yeah, and like the no membrane-bound organelles, just want to reiterate that bacteria and archaea both display prokaryotic cell structures. Also, bacteria use, I'm gonna write it on the side here since I ran out of space, but they use formal methionine as their initiator for amino acid to start protein synthesis. So we use methionine to start ours, and similarly in bacteria, it's formal methionine. Okay, let's look at Archaea. So unlike bacteria, Archaea do not have peptidoglycan or muramic acid. But they are similar in that they have circular chromosomes. And they differ again in their RNA, is in that it uses several different types of RNA polymerases across their species. Oh, can you hear me now? Oh, okay. Okay, glad you can still hear me. And then Archaea uses methionine as their initiator. And this is more similar to us than it is to bacteria, the fact that they use methionine for their initiator. And then let's finally look at eukarya. So, what can you guys tell me about eukarya? What are their key characteristics? What are their chromosomes like? Do we have double membrane, nucleus, or membrane-bound organelles? Yes, membrane-bound organelles. Great answer. Yep, that's right. Linear chromosomes as well. Do eukaryotic cells have a cell wall? Yeah, so we have eukaryotes. Comment says yes, out of cellulose, I think. Yeah, that's a great answer. So the plant cells have a cell wall, and it's made out of cellulose or chitin in certain insects. Comment says membrane-bound or bound cells, sexual and asexual reproduction, and multicellular. Yes, all great answers, and all of that is true. Can anyone tell me what the initiator amino acid is for eukarya? Yes, great answer, methionine. Any questions about this section so far? Okay, next we're going to look at different groups of bacteria, the six major groups, and what are the characteristics. Comment says UAG. Yeah, are you talking about the codon and the anticodon? Okay, yeah. So in the next section, we're gonna look at the characteristics that are used to place them into each of these six groups. I'm not sure how exactly to pronounce all these words, but I'm gonna try my best. It says, so the first bacterial group is the Firmicutes. So some of the characteristics that places bacteria in this group is that they need to be gram-positive, and that means they have the big peptidoglycan cell wall. Their DNA has low percentage of guanine and cytosine. They're usually rod or spear shaped. And some can photosynthesize, but some fix nitrogen. and or some use fermentation to get their energy. So there's lots of different ways that they can metabolize to get energy. And members of this group are known to cause anthrax, botulism, and tetanus, and other infections and diseases. But also, some of them are used to ferment milk products. Okay, next we're going to look at the Spirocales group, and these are known to be in corkscrew shape. And they have a flagella. And the flagella is usually within the outer sheath. And how these bacteria metabolize is through fermentation or sometimes by fixing nitrogen. And the types of diseases that this can cause are syphilis, and Lyme disease. Also, it's good to note that many of these bacteria are anaerobic. So bacteria that belong to this group are usually anaerobic. The next group is actinobacteria, and this is kind of the opposite of the first group that we looked at, in that they have a high percentage of guanine and cytosine, and they're gram-positive. And these are usually heterotropic and they're parasitic, so they're known to harm their host. And they cause tuberculosis and leprosy. And they're usually abundant in soil where they act as decomposers. The next group is calamideales, and they're spherical in shape and super small. These are also known to be a parasite, but they're also known to live inside the host cell for most part. These types of bacteria causes STIs like chlamydia, and they also are the most common cause of blindness in humans, which I thought was really interesting. And then the next bacterial group is cyanobacteria, and these usually live in, or they can live in colonies, but they can also live in their on their own. And these are very helpful for aquatic environments, so mostly found in aquatic environments. They're all photosynthetic. Some can fix nitrogen and they live in association with fungi, and when they do that, it's called lichens. And the last group, proteobacteria, and these have five major subgroups. So they have rod, sphere, or spherical shape. So they have all three types of, they come in all shapes. And they have a wide metabolic delivery, and they're known to cause dysentery, cholera, gonorrhea, ulcers, just a lot of bad diseases. And fun fact is that they're used to produce vinegar, and they can live in, these are the types of bacteria that you find to live in or on plant roots. And this helps the plants because they will fix the nitrogen and make the nitrogen in a form that's useful for the plants. So there's one more page, but I think this is a good place to stop since we're coming to the end. So let me know if you guys have any questions. I'm gonna put the link in the chat again for those who haven't gotten a chance to fill it out. So please fill out that form to get extra credit for the session, and I hope it was helpful. And um, one of the comments says, random question, do you know if there will be a session on week 10? So currently we are planning to have a session on week 10, but if anything changes, I'll have the professors make an announcement on D12. But as of now, yes, there will be a session on week 10. Also, if there's any specific topics that you might want to like see other examples of, feel free to request it from the professors and they can provide me with the material that would be most helpful for you guys. Especially if it's like around midterms week or finals week, you can ask them for things that might be the most helpful for you. Yeah, no problem. Thank you everyone for coming. Again, make sure to fill out the form so you can get the extra credit for coming.